Hello students, today we'll be speaking about gene libraries. For research purpose, for gene manipulation studies, it is necessary that we need to maintain the organism's DNA or genes in clonal libraries. And that is achieved by preparing a gene library. So what is a gene library? It is a collection of DNA fragments that represent the genetic material of an organism. There are two main types of gene libraries, genomic library and cDNA library. A genomic DNA library contains DNA fragments that represent the entire genome of an organism, including both coding genes and non-coding genes. Non-coding regions include introns, regulatory sequences. In this process, the DNA is usually cut into smaller pieces, cloned into vectors such as plasmids, bacteriophages, YARCs, and stored in host cells. Genomic DNA libraries can be retained for many years and propagated so that the copies can be sent from one research group to another research group. The process is usually done by constructing from the chromosomal DNA, if it is eukaryotes, where the nuclei is purified by digestion with protease and organic extraction. The derived genomic DNA is too large to get incorporated into a vector and thus needs to be broken up into desirable fragment sizes. Fragmentation of DNA can be achieved by physical method and enzymatic method. In physical method, mechanical sharing is usually preferred, which is done by using a narrow gauge syringe needle or sonication to break up the DNA into suitable size fragments that can be cloned. DNA fragmentation is random, which may result in variable size DNA fragment. In enzymatic method, it involves the use of restriction enzyme for the fragmentation of purified DNA. Partial digestion of the DNA molecule is usually carried out using known quantity of restriction enzyme to obtain fragments of ideal size. When we do with the enzymatic digestion process, the digestion of genomic DNA is preferably done in such a way we do with partial digestion. And one such enzyme which is used for partial digestion of DNA is SAW3A, that are generating the DNA fragments and are also compatible with the sticky ends produced by another enzyme known as BAMH1 enzyme. So if we are fragmenting the genomic DNA with SAW3A1, we can cleave the vector DNA by using BAMH1. Once the DNA fragments are produced, they are cloned into a suitable vector. The size of the genes and the organisms dictate which vector is good for holding the inserts. We can use lambda replacement vectors, cosmids, BACs, PACs, and YACs. When I speak about the use of bacteriophage DNA library, use of bacteriophage lambda as a vector, we cleave the bacteriophage lambda DNA with the same restriction nucleases that is used to cleave the genomic DNA. The two are mixed, DNA ligase is added, and the recombinant in the chimeric DNA is packed into phage capsids. Later, the phage capsid is allowed to infect the bacteria host. The recombinant phage multiplies as the bacteria multiply, generating millions of genomic DNA clones. The phages can be purified, and the collection of virion is called a phage genomic library. Genomic libraries usually are stored as phage particles in solution. This diagram speaks about the use of the bacteriophage lambda DNA vectors and the library of the human DNA. The human genomic DNA is partially digested with SAW3A that generate 20 kilobase fragment of the DNA molecule, where SAW3A gives usually a blunt end, but then we can make them sticky end by using adapters. On the other hand, we'll be taking your bacteriophage lambda DNA and we can cut them with BAMH1 by removing the non-essential region or called as replaceable region. And this also is generating your BAMH1 sticky ends. So being complementary, now when we mix your 20 kb fragment of DNA with lambda vector arms, that gives me the recombinant lambda DNA that can be packed into the phage capsids by in vitro phage assembly system. Later, we can run the infection to the bacterial host, and thus the library can be constructed. This speaks about the usage of cosmids as the vector. This picture speaks about the use of YARK vector to bring about the generation or construction of genomic DNA library of the human cells. How do we get to know the probable number of recombinants that can be obtained from a given organism? We have a formula which speaks about the number of recombinants can be calculated by getting the desired probability and also by finding out the fraction of the genome of one insert into the vector. So this speaks about how many number of recombinants can I get for an E. coli and how many can I get for a human genome. The advantages and the disadvantages are the advantage of genomic libraries. They help in the identification of clone encoding a particular gene of interest. It is useful for prokaryotic organisms having relatively small genomes. Genomic libraries from eukaryotic organisms are very important to study the genome sequence of a particular gene, including its regulatory sequence and its pattern of introns and exons. Disadvantages: genome libraries from eukaryotes have very large genomes containing a lot of DNA, which does not code for any protein, and thereby the non-coding DNA, such as replicated DNA and regulatory regions, which make them less ideal. Genomic library from a eukaryotic organism will not work. The screening method requires the expression of a gene. However, the application of genome libraries is very important because they determine the complete genome sequence of a given organism. They help in the study and generation of transgenic animals. They help in the study of the function of regulatory sequences in vitro. They help in the study of the genetic mutations. They are used for genome mapping, sequencing, and assembly of clone contexts, and they're useful for biodiversity and germplasm conservation. Thank you.